be talking to you today about, I know this is climate change and biodiversity, and I'm going to talk to you about a little bit different aspect of global change. And so this talk is mostly going to focus on urbanization, but as I'll explain in a minute, these problems are all interconnected. So my research uses urban environments as natural laboratories to understand the mechanisms of rapid evolutionary change. What do I mean by that? So one quick thing, the timer is not going on this, just so you know. Okay, so what do I mean by urban environments are natural experiments? So this is a picture of San Francisco. This could be a picture of New York City, Tokyo, even cities here in the UAE. And there are aspects of every city that are the same. Dominance of humans and buildings and impervious surfaces, fragmentation and loss of natural vegetative habitat. You have very small fragments of habitat like here, that there's some trees, but otherwise there's no trees here in this concrete jungle. And so with that, you get a complete shift in the biodiversity that can exist here, a complete shift in the nutrients that exist in this environment, and change in the abiotic and biotic properties. It's hotter, it's drier, it's more open. And so every city across the globe, you can find some of these repeated environmental changes that create kind of novel selective pressures. And so I'm not arguing that cities should be preserved for wildlife, but wildlife does exist in cities, and understanding how they are able to do that can both tell us something about how evolution functions, but also how life in the future of our planet is going to function. And so what I mean by that is this Anthropocene, this current geological epoch in which we live, is multifaceted. It's not just climate change. It's not just habitat loss and urbanization. It is the intersection of these novel range shifts like we just heard about, invasion fronts, novel distributions of species and species interactions, novel climatic regimes, and novel environments created by humans. And so my research really focuses primarily in this bubble here, the human effects on the structural and climatic environment in the areas in which we live. But as you see, I've drawn this as a Venn diagram because I also play around in this zone here, trying to use urban environments as space for time substitutions to understand climate change. And down here, the intersection of invasion and urbanization, trying to understand how humans facilitate range shifts. So I'm going to talk to you mostly today about anolis lizards. So anolis lizards are a fantastic model for evolutionary biology. If you're not familiar with them, they are the best model organism in my opinion, though many people do not consider them as model organisms. And the reason I say that is because in the eco-evo world, this is a classic adaptive radiation. There's over 400 species throughout Caribbean, Central, and South America. They have this repeated macroevolutionary pattern of diversifying into different environments with very strong trait-environment relationships that are very clearly understood. And so we can make predictions about their evolution on both short and long time scales to understand evolution in the wild. Not only that, but there's a wealth of genomic resources in the laboratory. We have CRISPR, we have cell lines. We have all of these resources that make this species the potential bridge between cellular molecular biology and ecology and evolution in the wild. I also love them because I think they're models for understanding rapid evolutionary change in the Anthropocene. I say that because they exist in the Caribbean, which is one of the most urbanized tropical regions consistently per square footage here. This is the island of Puerto Rico in the middle of the Caribbean. It's been colonized by Western society for 500 years. It is home to over 2 million people in a very small area. It was 95% deforested only 80 years ago. And you can see from this map of the Earth at night, it is just completely lit up with human impacts. And you can see this throughout the Caribbean too. On all of these Lesser Antillean islands, that's basically the entire island is dominated by urbanization. Not only that, this is a biodiversity hotspot. There's over 120 species of anolis just in here. And so understanding how these animals are dealing with this rapid and drastic evolution or environmental change can help us understand kind of more broadly how animals are dealing with this. And you probably have not really thought of urban environments as habitat for animals. Maybe some of you saw me get very excited to find geckos last night on a building. And if you've never looked for wildlife in urban areas, it's very common to think they don't exist here. They can't exist here. But in fact, if you know where and when to look for them, they're very common. On every single island I've gone to in the Caribbean, I have found at least one species of lizard that is not only existing in urban areas, but thriving in them. So this is also to give you a sense of some of that diversity and repeated adaptive evolution in this genus. These are all different species found across the Greater and Lesser Antilles that I've found in urban environments. And so I'm going to focus primarily on the species in Puerto Rico, and in particular, this one right here, Anolis chrysotellus. So my work uses a strategy of finding contrasting urban and non-urban environments in close proximity across a range of a species to try and understand parallel evolutionary change. And so this kind of natural laboratory setup then is that we have this parallel environmental change. So just like you have your treatments of petri dishes in the laboratory, each one of my cities is a treatment compared to the forest that is nearby. You can see some of those city pairs that I've worked in. So these are satellite images of some of my sites across the island of Puerto Rico. That map in the center, the white, is showing you the distribution of urbanization. So you can see it really is distributed across the island. And just looking at these satellite images, you can see that there's a lot of similarities between some variants, but in general, those urban sites are more similar to each other than the forest sites, and the forest sites are more similar to each other. And we can quantify this kind of in multi-dimensional environmental space to characterize that kind of environmental selective landscape. And so here's just a simple PCA of that environmental space, looking at things like canopy cover, impervious surface, light at night, openness of the habitat, temperature and precipitation. And what you can see is that the forest habitats all kind of move in the same direction away from the urban habitats down here. And so we do have this parallel environmental shift here repeated across the island. So now this sets up the question of, if we have parallel environmental change, and we know that this species is very responsive to environmental changes. There's been a wealth of research in this genus that has linked changes in the structural habitat, in the climatic habitat, and in the biotic communities to rapid morphological and physiological changes in only a couple generations. So with that background, we can then ask, what will happen in this natural experiment of urbanization? What kind of adaptive responses will we see? And can we detect, predict maybe even, how natural selection is unfolding at different levels of biological hierarchy, from the phenotypic all the way down to the genomic? So I'm going to start there with the phenotypic changes that we see in these lizards. So I mentioned that there was this wealth of research in this genus and that they're classic adaptive radiation. And so in Anolis lizards, we focus on two sets of traits that have been really key to understanding their adaptive radiation and that habitat specialization. So what I'm showing you here in this cartoon is the kind of classic Anolis drawing of this adaptive radiation, showing that on every island that these lizards colonized, they diversified into these repeated body forms, where you have lizards that live high up in the trees that are specialized for life up there with relatively short limbs and large toe pads, and then lizards far down on the trees with relatively long limbs, and this matches their ecological requirements. With that comes these traits that we focus on: the toe pads, So large toe pads high up in trees because the cost of falling is great. And long limbs down on the ground because they need to run quickly across the ground, whereas high up in the trees, short limbs beneficial because they need to hang on tight to those very thin twigs. We also see this diversification of, not yet, physiology. So we have these repeated body forms associated with the structural habitat, but we also see repeated physiological patterns where after there's this diversification of the structural environment, we then get habitat matching for the physiology. So you'll end up with a lizard that looks like this, that lives down by the streams and has a low thermal tolerance, and a lizard that looks almost identical but lives a couple hundred meters away up the side of the mountain slope where it's a slightly different environment. So what happens then in the urban environment? What might we predict is changing? So the first thing is that the urban environment is going to be structurally different than the forest environment that these animals evolved in. And so a lizard in this environment that's not able to navigate this type of structure is going to lose upwards of 50 to 60% of that habitat space. They're going to expend extra energy, their metabolic costs, injury costs, predation is going to be increased. And so all of that's going to create a selection against, or selection for being able to navigate this environment. I'm going to skip this video in the sake of time, but we also see, actually it's playing, so we'll go with it. We also see we also would predict that there would be a shift in physiological dysfunction in urban environments. So what I'm showing you here is my collaborator, Shane Campbell-Staton from Princeton, showing you how we test thermal tolerance in lizards. And so in increasing temperatures, we would predict that we would have a loss of muscular function in those hotter temperatures. So in urban environments then, what do we see? We have this prediction of broad open habitats, and so we can predict that there will be longer limbs. We have this prediction, this observation that there are these smooth surfaces that the lizards are clinging to, so we have this prediction that their toe pads will evolve to deal with that. Similarly, they're exposed to these hot urban heat island temperatures, and so we can predict that there will be an evolution of thermal tolerance. What do we see when we look at those phenotypes? Exactly that. So this is the beauty of anolis lizards. Because we have such a wonderful knowledge of those trait-environment relationships, they play very nicely with our predictions. And so we see here the forest animals, or the forest populations in green, the urban populations in white. I'm showing you here just the hind limb length and the size of the, that rear toe pad and the number of specialized scales on it. We see the same trend for a number of different traits. In general, what we're seeing is between a 3 and 5% increase in limb length and toe pad size between the forest and urban animals. And so these trait shifts were in line with what we predicted should enable them to function better in these habitats. Similarly, when we look at their thermal physiology, we would have predicted the higher body temperatures, higher function at higher environmental temperatures, and that's exactly what we see. So this is a metric called CT max. This is the metric of systemic dysfunction, or the temperature at which systemic dysfunction occurs. And what we're seeing here is that repeatedly across our urban sites in gray, we're seeing about a 1 degree Celsius on average increase in that maximum temperature that these lizards are able to tolerate. And that might not seem like a lot, but 1 degree Celsius can make the difference of being able to persist and find mates and pass on your genes versus needing to go and hide during the hottest part of the day. So now what you are all actually interested in is, how does this connect to genomics? Do we actually see evidence of these phenotypic adaptations at the genomic level? And I've done some work on both of those traits that I just told you about, the morphology as well as the thermal physiology. I went back and forth on which to show you today and decided to talk about the morphological stuff. But if you're interested in the genetic basis of thermal tolerance, we can also talk about that afterwards. So do we see evidence of urban adaptation at the genomic level? Well, first, just to give you that setup again, we went out, we sequenced paired populations across this island. We can say that they are actually genetically independent replicates because not only do we know there are deep genetic breaks in this species in particular across the island, but we did the population genetics just as a gut check and also to incorporate into our analysis. So this is a discriminant analysis of principal components of genetic variation. All this is showing you is each one of those points is an individual. They are colored by population, and you can see that there are clusters by region where the forest and urban populations in each region are more closely related to each other than they are to individuals from other regions. This is important because this actually establishes that replicated treatment that's an assumption of being able to test any ideas of adaptation and parallelism at the genetic level. Because if this were not the case, if, for example, we didn't have clear paired populations here, then we could possibly say, well, maybe any adaptation that we see repeatedly across populations actually just arose once and swept through all of them. But this is showing that, in fact, we do have clear genetic divergence across our treatments. So the first thing we can do then is ask kind of writ large, what is the genomic signature of natural selection in urban environments? What I'm showing you here is a Manhattan plot of the association, the genomic associations and divergence with urbanization. So this genotype environment association test is across all of our pairs of urban forest populations. And so it's not focused specifically on any one phenotype. What you might be noticing is we have just tons of outlier SNPs here. All of those SNPs in green are the ones that passed our threshold. We have no real clear peaks, although we do have these three on chromosome 1, 2, and 4. And what's interesting about these peaks is in a number of our other studies on thermal tolerance in urban environments, as well as divergence associated with xeric and mesic environments, we see repeatedly this very large peak on chromosome 1. So something very interesting is going on with that chromosome that we haven't quite figured out yet. When we look at what genes are in these peaks in particular, we can pull out a whole bunch of our top candidates here. When we look at what those do, they do things like immune function, cognition, metabolism, things that we know are changing in urban animals, things that we suspect should be changing, but also this is kind of a fishing expedition here, so I don't want to give that too much weight. It's for future to maybe kind of dig into more. The real power, though, of this, and why I'm showing this, is because now we can leverage the fact that we have this kind of differentiation associated with urbanization and map that back with our associations with phenotypes to actually try and understand what genetic changes are associated with our urban phenotypes. So getting back to those key traits, limb length and our toe pad morphology, we can then do a GWAS and ask what are the genetic associations across all of our animals. We have this incredible variation and very strong differentiation across our populations. And if you've ever done a GWAS, you might be saying, well, you need to have inbred lines and very big population effects of phenotypic change, and you would be right because you try and do this in a wild and a complex quantitative trait, and you get a lot of noise, right? No clear peaks, lots of outlier SNPs here, many genes. I mean, this is not unexpected. We expect that these traits are very complex and controlled by a lot of different genes. And this is also not dissimilar to what my colleagues have also found, trying to find the genetic basis of these traits. What we can do, though, is go back to that association with urbanization and try and tease some signal out of this noise. So the first thing we did was we looked at the overlap of outlier SNPs with the forelimb and hindlimb, the anterior and posterior elements of each of our traits. And this was kind of a step to just kind of narrow down our set of candidate genes. We could have skipped this step, but as you'll see in a second, we would have ended up with a lot more candidate genes. And so this was our kind of most narrow approach to identifying the most important genes underlying our urban adaptive phenotypes. So what I'm showing you here is on the x-axis is the posterior element for our limbs and our toe pad morphology, and on the y-axis is the anterior element. And so what I want to draw your attention to is the SNPs up here. And so these are the SNPs that are pleiotropic, that are being controlled by, or these are SNPs that are outliers associated with both the fore and hind limb. We can then leverage that strong and consistent urban phenotypic shift in order to kind of get at that genetic basis of this urban specific morphological adaptation by looking at the overlap of those that I just showed you, now with what we saw was changing across all urban populations. And when we do that, we can overlay now in gray, those are all of our, the gray diamonds are all of our outliers that were associated with urbanization, and what should be red diamonds, but are kind of oddly colored here, these are all of our SNPs that were identified across multiple analyses as being associated with both urbanization and phenotypes. And so these are our candidate genes underlying our urban associated phenotypic shifts. When we look at what those genes do, or we can identify those, we can then look at what those genes do, and we find that they are in fact associated with very relevant functions in human and in mice. We see that these genes that we identified associated with limbs have been implicated in disease states and limb development. We see that these genes associated with lamellae, or the specialized scales on the toes, that these are related to collagen and skin development. And the genes associated with the toe pad size are related again to keratin and skin development, as well as smooth muscle contraction, which is actually how those toe pads engage and disengage with the surface. And so what is cool here is we've actually identified candidate genes underlying these rapid adaptive shifts. What's even cooler though is when we kind of take a step back and look at all of those genes that are associated, not just this very narrow set, but all of these genes that are associated with urban morphological change, and we do a go-term analysis on it, what we find is that we hit on a huge, huge number of human phenotypes, and the majority of those are associated with disease states. Now, this was very surprising to us, and relevant disease states, right? Abnormality of skeletal morphology and limbs and skin development. And so what this suggested to us is that perhaps in humans, in mammals, tinkering with these very important genes has very bad effects. But in a different organism, in a different selective landscape, tinkering with these genes of very large effect can potentially have exactly the type of very large effect that could be adaptive. And so this is kind of opening our eyes about how we think rapid adaptation might happen at the genetic level, and what types of genes are being acted on by natural selection to produce these changes. Okay. In the minute I have left, I'm going to very quickly talk to you about this other aspect of using urban environments as a model for understanding climate change and invasion. And so this is research by my postdocs and graduate student in my lab. I'm just going to tell you 3 very quick stories, all related to the urban heat island effect. So the urban heat island effect is this idea that in cities you have increased temperatures even compared to only a couple kilometers away, and this is because of the way that the heat is radiating and absorbing within these environments. And so on the left here, this is some temperature profiles from one of my postdocs sampling in Puerto Rico. All of those red lines are urban sites, sampled across, I think, a couple of weeks, and all of the green lines are non-urban sites. And you can see that consistently in day and night, the urban environments are significantly hotter. I mean, upwards of 10 degrees difference between some of these sites. This is a pretty severe difference in the thermal environment that animals are experiencing. And so one of the things we're doing in the lab is trying to understand how there might be convergence in adaptation to the urban heat island across very divergent taxa. And so right now we just have phenotypic data, but each of these people are also digging into the underlying genomics, in some cases developing the genomic resources necessary, as Anthony is doing. He is currently in the middle of producing the first plethodon salamander genome. It is 25 gigabases, full of repetitive elements, and pretty ugly. But he has so far, with just the phenotypic data, shown that there is this consistent increase in the urban populations, convergent with what we saw in the lizards, where we have over here the urban salamanders are tolerating higher temperatures compared to the rural and the suburban animals. Once he has that genomic resource, he's going to start digging into what kind of genetic underpinnings there are. Similarly, Valentina, another postdoc in my lab, is looking at functional convergence in endotherms. So she's using hummingbirds and metabolic rate instead of thermal tolerance, and she, again, is finding these differences in that metabolic curve associated with urban heat. And then lastly, my PhD student, Fallon Meng, in collaboration with NYU Shanghai and an undergraduate there, have been looking at the evolution of thermal tolerance. So again, this is CT max, that critical thermal maximum, in an invasive species, the lanternfly. And what's cool about this, so I'm showing you here the CT max in 3 cities in the Northeast US, where this bug has been invasively spreading in the past 10 years only. It only arrived in New York, so Staten Island is in New York. It's only arrived there in the past 2 years. We see only in that short time period, though, a significant shift towards different thermal tolerance compared to its native range. And when I don't have a plot for this, but when we've compared the urban versus the non-urban within Shanghai, we see evidence of some sort of past selection on that thermal tolerance before the invasion even happened. So it's diverging within the native range, and again diverging in the invasive range after being bottlenecked. And so with that, I am pretty much out of time, so I'm just going to put up my conclusion slide and take questions.